Hello everyone, welcome to RDR Life Science. In today's video, we are going to discuss acid-fast staining procedure. Acid-fast staining procedure is a differential staining procedure. This procedure is used to stain acid-fast bacteria. These acid-fast bacteria are present in the genus Mycobacteria and Nocardia. Mycobacterium tuberculosis, which causes tuberculosis, Mycobacterium leprae, which causes leprosy, and many Nocardia species, which cause the disease nocardiasis, are examples of acid-fast bacteria. All these bacteria are stained using acid-fast staining. Acid-fast staining can be done using two different methods. The first method is Ziehl-Neelsen method. It is also known as Zn staining. And the second method is the Kinneon method. The basic difference between these two methods is that in Ziehl-Neelsen method, during staining, heat is used for stain penetration to bacterial cells. Since we are using heat in this method, it is also known as hot method. But in Kinneon method, there is no use of heat. Therefore, this method is known as cold method. In this video, we are going to learn how to do acid-fast staining practical using the Ziehl-Neelsen method. To perform this acid-fast staining practical, we need some apparatus. We need bacterial suspension, a sterile wire loop, clean glass slide, Bunsen burner. Then we need three different acid-fast reagents such as carbalfoxin, acid alcohol solution, and methylene blue. We also need wash bottle, a glass beaker with water, hot plate to boil water. We also require forceps, staining tray, and finally the most important is the microscope. Now we have all the apparatus required for acid-fast staining. Let's begin the practical. The first step is to make a bacterial smear on the glass slide. Take the test tube having bacterial suspension. Dip the sterile wire loop into the test tube and take out a loop full of bacterial culture. Now there is bacteria on the wire loop. Take a clean and grease-free glass slide. Place the loop containing bacteria in the center of glass slide and make a smear. So friends, after making smear, we need to fix this bacterial smear on the slide. Let the smear air dry for few minutes and then we are going for heat fixation of the smear. Hold the slide with the forceps and gently pass the slide through the flame for about 2 to 3 times. Please be careful and do not overheat the slide. Now the smear is fixed on the slide and the slide is ready for acid-fast staining. In acid-fast staining, we are going to apply three different reagents such as carbalfoxin, acid alcohol solution, and methylene blue in a sequence. Let's start. Keep the slide on the staining tray. The first stain we are going to add on the smear is carbalfoxin. It is primary stain. Take out carbalfoxin stain with a dropper. Flood the smear with carbalfoxin. Now hold the slide with forceps. We are going to apply heat to this slide. Take a hot plate and keep a glass beaker containing water on this hot plate. Once the water in the beaker starts boiling and steam generation starts, then keep the slide on the beaker. Due to the steam, the slide will get heated and this heat is essential for penetration of the stained carbalfoxin to the bacterial cells. Keep the slide on the steam for 5 minutes. After 5 minutes, now hold the slide with forceps. Keep the slide on the staining tray. Wash the excess stain on the slide with water. After staining with carbalfoxin, both acid-fast bacteria and non-acid-fast bacteria get stained with the carbalpoxin and appear red in color. The second reagent we are going to add on the smear is solution of acid alcohol. The acid alcohol solution acts as a decolorizer. Take out acid alcohol solution using the dropper and flood the solution on the smear and then wait for 30 seconds. After 30 seconds, wash the excess stain on the slide with water. The acid alcohol solution decolorizes non-acid fast bacteria. After decolorization step, non-acid fast bacteria lose the red color and become colorless, whereas there is no effect of acid alcohol solution on the acid fast bacteria. Acid fast bacteria retain the red color even after the decolorization step. The third reagent we are going to add on the smear is the methylene blue. Methylene blue stain acts as a counter stain. Take out methylene blue with a dropper and flood the smear with methylene blue. After the addition of methylene blue, wait for 2 minutes. After 2 minutes, wash the excess stain on the slide with water. Staining with methylene blue, only non-acid fast bacteria which are colorless get stained with methylene blue and appear blue in color, whereas acid fast bacteria doesn't take up the methylene blue and remain red colored. After washing, let the slide with stained smear to air dry for few minutes. Now the next step is observation of the stained smear on the slide using the bright field microscope. Keep the slide on the microscope and observe the smear under a 100x oil immersion lens. After acid fast staining, acid fast bacteria will appear red in color, whereas non-acid fast bacteria will appear blue in color. For more information on this topic, please read my blog post. You can search my blog post on the Google. In Google, type acid fast staining by RBR. You can see my blog post on the top written on acid fast staining method by rbrlifescience.com. You can also check your understanding on this topic by solving MCQ on acid fast staining procedure. Go to the end of the blog post. Below the solve MCQ on acid fast staining, click on start quiz and you can start solving the MCQs, which are very important for an exam point of view. So friends, thank you so much for watching this video. If you want to watch such more videos, then please like, comment, share and subscribe our YouTube channel RBR Life Science. For more detailed information, you can visit our blog rbrlifescience.com. The link to our blog website is given in the description. Thank you.